Good morning, children. We have already started with chapter number 5, Principles of Inheritance and Variations. And in that we have learned Mendelian genetics. We have studied about genetics, heredity, variations, then the types of variations, that is hereditary, and we have studied about Gregor Johann Mendel, father of genetics. We have studied more in detail about Gregor Johann Mendel, where he lived, where he was born, where he lived, and where did he studied. We have studied about rediscovery of Mendelism. Then we have studied about the 7 different characters that were selected by the Mendel with different traits for performing his experiments. We have studied about the terminologies in genetics, that is factor or genes, then locus, allele. We have studied about phenotype, genotype, phenotypic ratio, genotypic ratio, homozygous, heterozygous. And we have studied that why Mendel has selected only pea plant for his experiments, because of the various characters of the pea plant. Then we have studied about the Mendel's law of heredity, monohybrid cross, test cross, then deviations of Mendel's law, that is we have studied about the incomplete dominance. We have studied about codominance, in which we have studied the example of the blood group. Then we have studied dihybrid cross, test cross, chromosomal theory of inheritance. Sex determination was the last topic which we have studied. We have studied different types of sex determination. Children, in the description box, I have given the link of the chapter number 2, that is sexual reproduction in flowering plants, and the 5th chapter, principles of inheritance and variations, from where you will get the notes of these 2 chapters. Today we will start with linkage and recombination. Morgan and his group observed in Drosophila that when the 2 genes in a dihybrid cross were situated on the same chromosome, the proportion of parental gene combinations were much higher than the non-parental type. They attributed this due to physical association of the 2 genes and coined the term linkage to describe this physical association of genes on a chromosome, and the term recombination to describe the generation of non-parental gene combinations. Thus, linkage is a phenomenon of genic inheritance in which genes of a particular chromosome show their tendency to inherit together. Morgan and his group also found that even when genes were grouped on the same chromosome, some genes were tightly linked. That is, linkage is stronger between 2 genes if the frequency of recombination is low. This is shown in cross A, whereas the frequency of recombinations is higher if genes are loosely linked. That is, linkage is weak between 2 genes, that is cross B. Recombination of linked genes is by crossing over. Exchange of corresponding parts between the chromatids of homologous chromosomes is called crossing over. Now, linkage groups. All the genes linked together in a single chromosome constitute a linkage group. The number of linkage group in an organism is equal to their haploid number of chromosomes. This hypothesis was proved by T. H. Morgan by his experiments on Drosophila. Morgan and his group hybridized yellow-bodied and white-eyed females with brown-bodied and red-eyed males and intercrossed their F1 progeny, that is cross A. It was observed that the two genes did not segregate independently of each other, and the F2 ratios deviated significantly from 9:3:3:1 ratio. In F2 generation, parental combinations were 98.7% and the recombinants were 1.3%. In another cross, that is cross B, between white-bodied female fly with miniature wing and a male fly with yellow-bodied and normal wing, parental combinations were 62.8% and recombinants were 37.2% in F2 generation. Thus, it was proved from the crosses that the linkage between genes for yellow body and white eyes is stronger than the linkage between the white body and miniature wings. Now, chromosome maps or linkage maps. Alfred Sturtevant, that is Morgan's student, used the frequency of recombination between gene pairs on the same chromosome as a measure of the distance between genes and mapped their position on the chromosome. Genetic maps are now used as a starting point in the sequencing of whole genomes as done in case of Human Genome Sequencing Project. Now, we will start with the new topic, and that is mutation is a sudden, stable and inheritable change in genetic material or DNA sequences of an organism. The organism which undergoes mutation is called mutant. The chemicals and physical factors that induce mutations are called mutagens. Example, UV radiations. Mutations are of different types like gene mutations and chromosomal mutations. Gene mutations are further of two types, that is point mutation, frame shift mutation. Whereas chromosomal mutations are further divided into structural variation aberrations and numerical variations. Further, numerical variations are of two types, that is aneuploidy and polyploidy. And aneuploidy is further divided into monosomy and trisomy. Polyploidy is further divided into autopolyploidy and allopolyploidy. Point mutation arises due to change in single base pair of DNA. Frame shift mutation refers to deletions and insertions of base pairs in DNA. Point mutation arises due to change in single base pairs of DNA. Frame shift mutations refers to deletions and insertions of base pairs in DNA. The loss or gain of a segment of DNA results in structural alteration in chromosomes because genes are located on the chromosomes. The alteration in chromosomes result in abnormalities and is common in cancerous cells. When the members of a homologous pair of chromosomes fail to segregate during meiosis, aneuploidy occurs. It means there is loss or gain of one or more chromosomes. Due to this, there may be monosomy, that is, lack of one chromosome of normal complement, or trisomy, that is, 3 instead of normal 2 chromosomes. Polyploidy occurs when there is failure of cytokinesis after telophase stage of cell division, resulting in an increase in a whole set of chromosomes in an organism. In this phenomenon, the cell has loss or gain of 3, 4 or more sets of chromosomes. Further, autopolyploids are polyploids with multiple chromosome sets derived from a single species. And allopolyploids is where chromosomes are derived from different species. That is, result of multiplying the chromosome number and forming a hybrid. Now let us study about pedigree analysis. The analysis of traits in several generations of a human family in the form of a family tree or diagram is called pedigree analysis. It provides a strong tool which is utilized to trace the inheritance of a specific trait, abnormality, or disease. Inheritance pattern of traits in human beings cannot be studied by crosses as in case of other organisms due to certain reasons. And that reasons are: the progeny produced is very small, usually one, and therefore takes long time. Second is, controlled crosses cannot be performed. The symbols used in pedigree analysis are given here, that is male, square for male, round for female, then diamond shape for sex unspecified, then the shaded one for affected individuals, then the line between the square and circle shows the mating, then 2 lines between the square, male and female, shows the mating between the relatives, then these are the parents, above are the parents, and below shows the progeny, right? Then the monozygous twins and the non-identical twins, identical and non-identical, that is monozygous and dizygous twins. Okay, and if the offsprings shows the shaded one, then it means the offsprings are affected. It shows affected offsprings. Now let us study about genetic disorders. These are disorders or illness caused by one or more abnormalities in the autosomes or sex chromosomes of the person, thus referred to as autosomal disorders or sex-linked disorders respectively. The genetic disorders are divided as Mendelian disorders and chromosomal disorders. Examples of Mendelian disorders are hemophilia, cystic fibrosis, color blindness, sickle cell anemia, thalassemia, phenylketonuria, whereas the chromosomal disorders examples are Down's syndrome, Turner's syndrome, and Klinefelter's syndrome. Let us study about the Mendelian disorders. These are mainly determined by alteration or mutation in a single gene. These disorders are transmitted to next generation according to the principle of inheritance and can be studied by the pedigree analysis. This can be dominant or recessive like autosomal dominant, example Huntington disease. Autosomal recessive like sickle cell anemia, cystic fibrosis, thalassemia, etc. Then sex-linked dominant, example defective tooth enamel. Sex-linked recessive, example color blindness, hemophilia, etc. And some Mendelian disorders like hemophilia, which is a sex-linked recessive disease which is transmitted from an unaffected carrier female to some of the male offsprings. Due to this, patient continues bleeding even to a minor injury because of defective blood coagulation, thus also called bleeder's disease. The gene for hemophilia is located on X chromosome. In this disease, a single protein, that is a part of cascade of proteins, means group of proteins involved in blood clotting is affected. The possibility of a female becoming a hemophilic is extremely rare because mother of such a female has to be at least a carrier and father should be hemophilic. Example, females suffer from this disease only in homozygous condition, that is XCXC. The family pedigree of Queen Victoria, who was a carrier of hemophilia, shows a number of hemophilic individuals. Now, let us study about color blindness. It is a sex-linked recessive disorder which results in defect in either red or green cone of eye. It does not mean not seeing any color at all. In fact, it leads to the failure in discrimination between red and green color. The gene for color blindness is present on X chromosome. It is present mostly in males XCY because of the presence of only one X chromosome as compared to 2 chromosomes in females. A heterozygous female has normal vision but is a carrier and passes on the disorder to some of her sons. Now the types of color blindness: monochromatism, the condition of complete color blindness in which all colors appears as shades of gray. Dichromatism, the condition of partial color blindness in which only 2 colors are perceptible. Example, red, green, blue, yellow. Daltonism, the condition of color blind person who is unable to identify red, green color. Hemophilia and color blindness shows crisscross inheritance pattern where inheritance of sex-linked character is transmitted from father to daughter or from mother to son. Protanopia, the color blindness characterized by defective perception of red and confusion of red with green or bluish green. Deuteropia, a form of color blindness characterized by insensitivity to green color. Now let us study about sickle cell anemia. It is an autosomal linked recessive trait that can be transmitted from parents to the offsprings when both the partners are carrier for the gene, that is heterozygous. This disease is controlled by a single pair of allele, HBA and HBS. Only homozygous individuals for HBS, that is HBS show the diseased phenotype. The heterozygous individuals are carriers, HBA, HBS. The amino acid composition of the relevant portion of beta chain of hemoglobin and forms a normal individual. It is caused by the substitution of glutamic acid by valine at the 6th position of the beta globin chain of the hemoglobin molecule. The substitution of amino acid in the globin protein results due to single base substitution at the 6th codon from GAG to GUG, which is also an example of point mutation. About codons and amino acids you will be studying in next chapter. The mutant hemoglobin molecule undergoes polymerization under low oxygen tension, causing the changes in the shape of the RBCs from biconcave disc to the elongated sickle-like structure. Its gene is located on 11th chromosome. This defect is beneficial to people living in malarial prone areas. Now let us study about the chromosomal disorders. These are caused by the absence, excess or abnormal arrangement of one or more chromosomes. The chromosomal disorders can be studied by the analysis of karyotypes. A karyotype is an organized profile of a person's chromosomes according to their shape, size and number. Let us study about the Down's syndrome. It was described by J. Langdon Down in 1866. It occurs due to the presence of an additional copy of chromosome, that is trisomy of 21st chromosome in humans. It is also seen in chimpanzees and other related primates. Symptoms of Down's syndrome are: individuals are short-statured with small round head and furrowed tongue and partially open mouth. Palm is broad and characteristic palm crease. Slow mental, physical and psychomotor development. There are some other chromosomal disorders like Turner's syndrome. It is a disorder which is caused due to the absence of one of the X chromosome, that is 45 with X0. Symptoms of Turner's syndrome are: affected females are sterile as ovaries are rudimentary, that is not very well developed. Lack of secondary sexual characters and poor breast development. Short stature, small uterus, puffy fingers and webbed neck. Another chromosomal disorder is the Klinefelter's syndrome. It occurs due to the presence of an additional copy of X chromosome, resulting in the karyotype of 47 XXY in males or 47 XXX in females. The extra inactive X chromosome in karyotype of Klinefelter's syndrome is called the Barr body. Thus, XXY individual have single Barr body, whereas XXX individual have two Barr bodies. Symptoms of Klinefelter's syndrome are: individuals have masculine development but feminine characters like development of breast, poor beard growth, and feminine pitched voice. Such unclear individuals are sterile. Along with this, there are some other Mendelian disorders also, like thalassemia, cystic fibrosis, phenylketonuria. Let us discuss about these three Mendelian disorders in brief. Thalassemia is an autosomal recessive disorder which occurs due to either mutation of genes resulting in reduced rate of synthesis of one of the globin chains of hemoglobin. Anemia is the main feature of this disease. There are two forms of thalassemia, that is alpha thalassemia, production of affected alpha globin chain which is governed by genes on 16th chromosome, and beta thalassemia, production of affected beta chain which is governed by a gene on 11th chromosome. Phenylketonuria, it is an inborn error of metabolism which is inherited as an autosomal recessive trait. Its gene is associated to 12th chromosome. It is a rare disease in which individual lacks an enzyme called phenylalanine hydroxylase, which is needed to break down an essential amino acid phenylalanine into tyrosine in liver. This phenylalanine is accumulated and gets converted into phenylpyruvic acid and other derivatives, leading to mental retardation due to accumulation in brain. These are also excreted through urine because of its poor absorption by kidneys. Now, cystic fibrosis. It is the most common lethal genetic disease due to a recessive mutation on the 7th chromosome. The body produces abnormal glycoproteins which interferes with salt metabolism. That is all for today. I hope you have understood the topic. With this, we have completed the chapter. Thank you.